system. What does he actually do? And whenever there was obviously my personal gut feeling. I'm not saying that it was correct, but whenever there was a function of the brain that people didn't know where to put and where to locate, they would put it in the limbic system. Oh yeah, that's a function of the limbic system. That's how it works. That is obviously not a correct impression, but it gives you an idea because the limbic system is extremely complicated. It does explain, it's essentially involving a number of functions, and these functions are very interconnected between each other, and therefore it's actually very difficult to define the limits of each pathway, each anatomical structure, and its function. So I'll try to make it as clear as possible, as I said. So let's start. Let's jump into it. So first of all, I'm going to talk about a broad definition and historical definition of the limbic system. Then we're going to talk about the structures from an anatomical perspective. Where are they and how are they shaped, and what are their anatomical relationship? Very briefly, because there are so many that actually we're not going to touch all of them. Then we're going to talk about the pathways, and that is actually the core. This time around, as opposite to last time, this is going to be the core of my lecture, because I'll try to explain to you what is the actual function of the limbic system. And then I'll close the presentation, put in some references, and obviously answering your questions. So what does limbic mean? So it's called the limbic system because it's at the edge of the two hemispheres. What does it actually mean? If you take a cadaveric brain and you cut it in half, you will see that the limbic system is sitting literally at the edge between, the subcortical space and the basal ganglia and the thalamus. Fact is that actually the basal ganglia and the thalamus are also involved in the limbic system, and the hypothalamus is also involved in the limbic system, but with different functions than the ones you've probably seen in previous lectures from my other colleagues, or at least part of those. It's a very ancient system. It's constant in every mammal, and it probably dates back to reptiles and dinosaurs as well, which makes it even fascinating. And it has been object of various study. The first one to use the word limbic was Paul Broca. You might have heard it for the Broca area, and also the diagonal band of Broca, which is another structure that we're going to meet today. Then it was further defined by James Papez, who defined the Papez circuit, which is responsible for memory and emotion processing, but mostly memory. And then finally, he essentially retained the name limbic because Paul MacLean renamed the whole system as a limbic circuit and defined the Papez circuit as a part of the limbic system. Now, let's name some structures. As I said, there is a huge list of structures involved in the limbic system. These are some of the gray matter structure. I would say the main ones are the amygdala, the hippocampus, and all the structures around these two. So the parahippocampal gyrus, the hypothalamus, the dentate gyrus, the cingulate gyrus. And there is huge amount of white matter tracts as well. Main ones is the fornix, but there's also stria terminalis, stria medullaris, ventral amygdalofugal pathways, fasciculus retroflexus, loads of them. We're going to mention pretty much all of them. I'm going to try to explain where they are. I would say that for a medical student level or exam, asking in great details the limbic system is kind of a cruel question because, as I said, it's very difficult. It's rich of structures and connection, but also, to be very fair, it's also a system where we don't actually know everything about it. We really don't, and many of the theories and the pathways are still under investigation by several groups across the world, in animals and in humans. So, first of all, from a general perspective, what does it actually do? Well, there are 4 functions that I would say pretty much everyone should remember, which are memory. And we talk about this. The Papez circuit is probably the main pathway involved in memory. Behavior and emotion, those are very closely related, and there are so many pathways related to behavior and emotion as well. But this is what part of the limbic system does. And then, weird enough, olfaction. So the sense of smell, is very closely related to the limbic system, and there is an evolutionary theory behind that, and we're going to see why. So let's start talking about a bit of topographic anatomy. So we've seen this picture on the previous lecture. We're not going to focus on the hemispheres because we need to come back to that. But let's focus on the center of the brain. As you might remember, there is this area which is defined as the central core of the brain. Laterally, the entry point of this area is the insula. Okay, medial to the insula, you got the extreme capsule, the external capsule, and then the putamen pallidus and the hypothalamus, really, in this order. Now, let's see what's around the hypothalamus. Well, around the hypothalamus, you have, on the top of it, you got the lateral ventricle, which in this particular section, which is exactly into the interhemispheric space, you got the septum, on top of which you got the corpus callosum. Around the corpus callosum, as we've seen last time, we got the cingulate gyrus. Now, the cingulate gyrus, between the corpus callosum and the cingulate gyrus, there's a very tiny layer of gray matter, of neurons, which is actually a vestigial organ. It was probably more developed in reptiles. Its name is called the indusium griseum. You don't really need to remember this. It's just to remember that there is this layer here. This indusium griseum becomes bigger and bigger and bigger here, thicker, I would say, becoming the dentate gyrus, which enters the hippocampus here. And we're going to see it's actually very important. It's a very important structure in the hippocampus. Anteriorly to the cingulate gyrus and the corpus callosum, if you remember, there are a couple of gyri that are very important. They're not very important per se, but they're important as anatomical landmark. So remember, the third ventricle on both sides is covered, essentially is surrounded by the hypothalamus. The hypothalamus is literally here, is the wall of the third ventricle. Anterior to the hypothalamus, you get a region called the septum, which is super important for the limbic system. And then anterior to it, you have this paraterminal gyrus and the paraolfactory gyrus. Now, these are not particularly important for the limbic system, but deep inside this gyri, you have the apex of the putamen and pallidus, and anterior to that, you have the septal nuclei. Inferior to the septal nuclei, you have a couple of nuclei that are the nucleus accumbens and the nucleus basalis of minor. They're also involved in the limbic system. And this is just a cadaveric specimen to confirm what, to just resume what you say. This is the septal region. This little gyrus here is the paraterminal gyrus and paraolfactory gyrus. Deep inside here, nucleus basalis, septal nuclei above, nucleus basalis and nucleus accumbens here. We're going to see this in great details. Now, we mentioned the hippocampus. Now, I suspect that in the lecture about the temporal lobe, you guys had a lot of information about the hippocampus. So I'm just going to try to make a very brief summary about this. So the hippocampus, this is seen from the medial side of the temporal lobe. So this is the base of the temporal lobe. The eyes and the olfactory area is here, on top of which you have the basal ganglia. This is the frontal lobe. This too is the olfactory nerve. And this area is the hippocampus. Now, the hippocampus, as you can see here, consists in a gyrus that goes all around the hippocampus, which is called the parahippocampal gyrus. On this gyrus, at some point, you got a population of neurons, which are marked with asterisks here, which is called the entorhinal area. Okay, this is part of the hippocampus. Now, the hippocampus tends to rotate and fold on itself, essentially. And what you can see here is that on top of which there is a very small band of white matter, which is called the fimbria. The fimbria is the actual output of the hippocampus. It becomes thicker and thicker and thicker, and enters two very important white matter structures, which are the fornix, the 7, and the stria terminalis, which wraps around the thalamus, together with another white matter bundle, which is called the stria medullaris. Oh, there was an animation here. I forgot about this. Anyway, those are the main points. This is just for a brief recap, and also to tell you about another name that you might encounter studying this area. The combination of the hippocampus, the actual hippocampus, which is called the uncus or cornus ammonis, this area here, which is the actual body of the hippocampus, and its parahippocampal gyrus, is called the piriform lobe, because it resembles kind of a pear, if you want. Hippocampus is called hippocampus because it resembles a seahorse. Hippocampus is the Latin name for seahorse. So if you counted the term piriform lobe, this is what it is. Now, let's have a look at the section of the hippocampus in a bit more details. The hippocampus is extremely important for memory, and we're going to see the pathway of the hippocampus later on. Now, the hippocampus is one of the few regions of the brain, it's probably the only region, I might be wrong on this, but definitely one of the main regions of the brain that is made of what is called archicortex. So it's a very ancient kind of cerebral cortex, which is constituted essentially by 3 layers, really. And the reason why this is important is because from an evolutionary point of view, this is a very old cortex that is being preserved since the age of essentially the first mammals, mice, literally, or probably even before, even, particularly evoluted dinosaurs, such as raptors, for example, they probably had this already. Now, what is important to remember? So this area here is actually facing the interpeduncular system and the tentorial notch, while this area here is a bit more lateral. You're going to see a number of very confusing schemes about the hippocampus. Sometimes the parts are inverted, but what you need to remember is the main population of the hippocampus are these pyramidal cells, and essentially named C1 from here, C2, C3, and C4. Before this population of pyramidal cells sitting here, you got the subicular cortex, which is also made by pyramidal cells, but with multi-layers. Now, the C1 sector is the one more sensible, more vulnerable to hypoxia. When you have a concussion, a brain concussion, this hits the tentorial notch, you have hypoperfusion of this area, and you got amnesia, retrograde or anterograde amnesia. And that's why, because these cells are extremely sensible even to very transient hypoxia. All these cells receives input. Let me try to, okay, I can't really move the camera, whatever. All these cells receives input from the entorhinal cortex through a couple of inputs, which are the entorhinal perforant input and the alveolar pathway. They also receive inputs from the dentate gyrus, which kind of is enveloped around these cells from the area which has been before a bit more closer to the fornix and the fimbria, up to the head of the hippocampus. What is important also to remember is that this is called subicular cortex, and this scheme is actually quite deceiving. Let me go back to this for a second. What is the subiculum? So as we say, this is the parahippocampal gyrus. The superior portion of the parahippocampal gyrus is called subiculum. You can see it here as well. Okay. So now, just for a summary, inputs from the entorhinal cortex and the dentate gyrus go into this C1, C2, and C3 and C4 cell. Sorry, CA1, CA2, CA3, blah, blah, blah. CA because cornus ammonis. Cornus ammonis is the head of the hippocampus. That's why they're called CA1, CA2, and CA3. So these inputs from the entorhinal cortex and the dentate gyrus reach these cells. These cells, they merge their axons. They send axons back to the entorhinal cortex, but mostly they send axons onwards to the fimbria. They merge together. They become more and more and more. They enter the fornix, and then they go through that pathway that we're going to see in a minute. Here we go. So this is another section seen laterally. So you can see the lateral ventricle here. This is the temporal horn of the lateral ventricle, and this is the head of the hippocampus. So we were looking at the hippocampus medially earlier on. Now we're looking at it laterally. If you can see the head of the hippocampus, there is this very clear white matter structure here that starts from the end of the hippocampus and goes back. And then it turns upwards, emits another one on the other side, and this is essentially the commissure of the fornix. So this whole structure, C-shaped, is the fornix. It's a massive white matter structure, very interesting, turns upwards and then anteriorly, is at the bottom of the septum between the two lateral ventricles, and then anteriorly it goes down and laterally and ends up into a couple of nuclei that are the mammillary bodies. As I said, we're going to see the pathway entirely. What I want you to remember here is the spatial relationship between the fornix and the ventricles. It starts on the lateral horn of the temporal ventricle, then it goes backwards, meets the other fornix on the other side, and runs at the bottom of the septum, at the medial side of the lateral ventricle, the frontal horn this time around, and then goes anteriorly, laterally, and ends up in the mammillary bodies. From a topographical point of view, this is quite important because the fornix is a structure that you actually see surrounding the foramen moro. It's both lateral, medial and lateral to it because of the course that I mentioned earlier on. So this is another view from the top. You can see the head of the hippocampus here, the fornix running around here, becoming the body of the fornix, and then laterally turns down and surrounds the foramen motor. Here's the access to the third ventricle. This is a lateral ventricle. This hole is jumping into the third ventricle. You can see the choroid plexus here. What is also interesting is to see the thalamus. Around the thalamus, in this area, you have a white matter structure which is called the stria terminalis. We're going to see why this is important. On top of which you got a coded nucleus. Now, this is just a recap slide to show you what I've told you on a DTI, so on a tensor imaging MRI scan. What is also very interesting about this specific dissection is that you can see the end of the fornix here. So the fornix, this is the back, this is the anterior portion of the fornix, this is the body of the fornix, turns laterally, ends up into this little lump here on the bottom of the third ventricle, which is the mammillary body. On the mammillary body, there's a white matter bundle which goes into the thalamus, mammillothalamic tract. We're going to review all of this in a moment. Another very important structure is the amygdala. So we're back to the medial view of the temporal lobe, and in green you can see essentially the amygdala in transparency. You cannot see it on the surface, as we seen before. Parahippocampal gyrus, head of the hippocampus, this is the cornus ammonis or uncus, and then you have the amygdala embedded inside the this portion of the parahippocampal gyrus. On an MRI scan, you can see it's sitting here essentially. This is the head of the hippocampus. This is the amygdala. Eyes, beginning of the frontal lobe, lateral temporal lobe. The amygdala is sitting right here. And if you take a basal view of the amygdala, means almond because it's almond shape. It's a nucleus which is almond shape. This is the basal temporal lobe. Part of it has been removed. You got the body of the ventricle at this point, and here's the head of the hippocampus, sorry, and on top of which, well, anterior to it, to which you can see with the asterisk the amygdala. The amygdala in itself is divided in several subnuclei. There are every now and then someone adds a nucleus to the amygdala, but essentially there is a central nucleus, a medial nucleus, and then a basolateral nucleus. You don't really need to remember that. Just remember there is a very complex nucleus divided in several subportions. Another important structure is the thalamus, which is another important structure involved in the limbic system is the thalamus. There is an anterior nucleus and there is a mediodorsal nuclei. These are the main nuclei involved in the limbic pathways. They're also divided in further subnuclei. Specifically, there is a anteromedial, anteroventral, and anterodorsal anterior nucleus of the thalamus, and then you have a magnocellular, parvicellular dorsomedial nucleus of the thalamus. This is just for your curiosity. You don't really need to remember that. The main take-home message is anterior nucleus of the thalamus, mediodorsal nucleus of the thalamus are involved in the limbic system, and we're going to see how. Now, let's jump into the main core of this lecture, because as I said, from a surgical point of view, the limbic system is really a very spread out system. You don't really have really have a single point where on the brain where you can see the limbic system, as you're probably seeing now. There are a number of structures all in connection with each other, and they are responsible for a number of functions. So this is what we're going to see now. We're going to see in details what these structures are and what these pathways do, essentially. Now, funny enough, we start with the olfactory system, which is actually the simple one. Now, the olfactory system technically is a somatosensory system. It's one of the systems that we use to actually feel the external environment. But is really closely related to the limbic system. And we feel that there is a very important evolutionary reason for that. An evolutionary biologist can probably explain to you this way better than me. But this is what you can see on the left-hand side is a calc, essentially as a cast, sorry, of the intracranial compartment of a T-Rex, of a dinosaur. And you can see how developed was the olfactory bulb and the close contact that it had with the cerebral hemisphere. And these are schemes comparing essentially the development of the cerebral hemispheres and the relationship they have with the olfactory system. Why the olfactory system is so important for the limbic system? Well, because from an evolutionary point of view, the visual system developed later. So many animals nowadays as well, they actually haven't got a visual system as developed as ours. Some others do. In fact, it's actually even better than ours, like, for example, lions, big predators such as lions, leopards, tigers. But many others, like cats, dogs, they haven't got such a refined visual system as we do. And many animals are colorblind. The olfactory system was effectively the very first system that most of the animals use to move around their environment. And essentially it evolved together with memory, because these animals were hunting or avoiding predators, so they needed to smell predators from great distance, or to smell prey from great distances, and remember where the prey were, or where they were going, trace them back somewhere or another. So there was a strong connection with the olfactory system and the memory, and also the decision-making of this animal. And that's why you have this very closely related connection between the olfactory system and the limbic system. It's been preserved up to nowadays, literally, and we have this connection ourselves. If any one of you has read a book called The Research of Time Loss, I think that's, I remember the French name, I can't remember the English translation, but essentially it's a book by Marcel Proust, a French author. He always, there's a specific episode when he walks around the street and there's a smell hitting his nose, and he remembers when he was a kid. That is something that it might happen to you continuously every now and then. You feel a smell and you remember something that you haven't remembered for a very long time. That is why, because there is an extremely strong connection between the olfactory system and the limbic system. So let's analyze the pathways of the olfactory system and why it's related to the limbic system. So the olfactory information enters your nose, of course, affects your receptors, and then your receptors are in continuation with the olfactory bulb, which is lying into the cribriform place. The olfactory bulbs sends information to the olfactory nucleus, and from here the olfactory nucleus sends a number of information to a number of structures. The most important ones are the entorhinal area, which we are seeing, which is in connection with the hippocampus, the amygdala, which is in connection with a number of other structures, we're going to see them later on, the septal nuclei, and the orbitofrontal cortex. Now, when you actually perceive the smell, the orbitofrontal cortex and the entorhinal areas are the two activating. These other pathways are involved in different functions that we're going to see later on. So let's keep it aside at the moment, but just remember there's a connection between the olfactory system and the area of the abenula, which is sitting in close proximity to your pineal gland, and there is a close connection between the olfactory system, the hippocampus, and the amygdala. If you want to see it in a bit more graphic way, this is what I'm gonna do from now on. I'm gonna show you the skin, and then I'm gonna show you some images that can explain the concept a bit better. This is your olfactory bulb. This is where you have one descending and one ascending pathway, and essentially the ascending pathway is a bundle of white matter called the medial olfactory stria, while the descending pathway is the lateral olfactory stria. And this is the connection with the amygdala and the head of the hippocampus. And here you got the connection with the septal region and the orbitofrontal cortex. And here you got a beautiful dissection from this paper. I would strongly recommend to read it. You have a basal view of the brain. Part of the temporal lobe has been removed. You have here the gyrus rectus that we mentioned last time. Here's the orbital gyrus of the frontal lobe. Here's your olfactory bulb. And you can see the lateral olfactory stria, beautifully dissected here in close connection with the amygdala here. There is also a bit of dissection of the uncinate fasciculus, which is connecting the temporal lobe with the frontal lobe. This is just to show you how close this is the connection between these structures. From here, there are several white matter bundles connecting the olfactory system with the head of the hippocampus and the entorhinal cortex as well. The head of the hippocampus is sitting here, has been removed in this particular specimen. Next circuit is the pathway is the Papet circuit, which is the main memory pathway. Now, this one is something that is responsible for, is literally your brain CPU. That's what it is. So memory processing, that's what it does. Now, this scheme sounds very complicated, but I'm actually sure that you heard about this pathway one way or another during your medical school training. It's essentially a circle pathway that tends to repeat itself over and over again. if your brain needs to actually store this information somewhere. If you don't need to store this information, this pathway at some point stops working for that particular information, and the information gets discarded. So what I want you to focus on is the hippocampus. So the hippocampus is really the core of this pathway, is literally the CPU of the brain. As we said before, receives information from the entorhinal cortex and the dentate gyrus. There is also a direct connection between the entorhinal cortex and the CA cells, but I forgot to put it here. Where the information from the entorhinal cortex and the dentate gyrus come from? Well, pretty much from the whole rest of the brain, but mainly from an area of the brain that is called the posterior association area. Essentially, is an area of the inferior palatal lobule when you get spatial, visual information and auditory information to process. Direct connection with the auditory areas of the temporal lobe, some connection from the central area, some connection from the brainstem, pretty much everywhere, as you've seen. And also, some of this connection, they go to the parahippocampus gyrus before entering the entorhinal cortex, specifically the subiculum. But anyway, entorhinal cortex, dentate gyrus, hippocampus. The hippocampus sends this information through the fimbria and through the fornix to the to the mammillary bodies. The mammillary bodies then send through the mammillothalamic tracts this information to the anterior nucleus of the thalamus. The anterior nucleus of the thalamus send it to the cingulate gyrus. The cingulate gyrus send this back through the dentate gyrus and through the subiculum and the parahippocampal gyrus to the entorhinal cortex, and goes on and on and on again. If you need to remember a fact, a number, semantic memory, something that you have learned, you will actually activate this circuit, this pathway over and over and over again, until at some point the cingulate gyrus will send this information to the prefrontal cortex and to the very spread out network of the long-term memory. We think that is located in between the cingulate gyrus and the prefrontal cortex, and that information is not going to be lost anywhere near in the future. If this information is not important, the cingulate gyrus, which is involved in decision making, and we're going to see it later on, the information is discarded. I don't care about this, forget about it. Typical example, remember a random number, 54650. If I ask you this number in 10 minutes, you train yourself to remember it. This circuit will be activated and you will be able to remember it. If I ask you this number tomorrow, probably you would have forget about it. And that because this pathway has decided that it's not important for your daily lives. Let's have a look from an anatomical perspective. This is a summary of what we just said. So head of the hippocampus, fimbria, crucis of the fornices, body of the fornices, go down and laterally, mammillary bodies, mammillothalamic tracts, anterior nucleus of the thalamus, cingulate gyrus. This scheme is taken from Kandel, which is one of my favorite neurophysiological books. What it shows you is that memory, obviously there are many types of memory. What is important to remember about this pathway is that this is a pathway that is activating what is called the explicit declarative memory. So when you learn something, when you need to remember a notion, that's the pathway that you actually use. Implicit non-declarative memory, such as, for example, learning procedural skills, learning how to play an instrument, how to operate, how to do anything manual, driving, for example, is performed by different structures of the brain, specifically by the basal ganglia. They probably talked about it my colleagues probably talk about it in their lecture. So this is the memory pathway of the limbic system. Now let's go to the emotional behavior pathways. We are going to consider them together. Now, this is probably the most complicated part, and also is relatively unknown still, in the sense that some of the information that I'm giving you might not be completely precise, because I'm not a neurophysiologist, I'm afraid. And some of the notions that I'm giving you are not precise also, not because of my ignorance, which definitely is big, but because they're not completely clarified from a scientific point of view. We haven't got enough information as yet. We got many studies on animals, not many studies on humans to actually define these pathways. So it's so complicated that I actually divided this portion in two portions. The first portion is related to the inputs of this system, and the second portion, which is the longest one, is related to the outputs. The core nucleus of this system is the amygdala. So the amygdala is responsible for emotion processing and a number of other functions that we're going to see in a moment. These are the inputs of the amygdala. A bit like the hippocampus, it receives inputs from pretty much everywhere else in the brain. So, posterior association area of the parietal lobe receiving visual information, somatosensory information, and visual information sends this information to the amygdala through the external capsule, the extreme capsule, and the inferior longitudinal fasciculus. The amygdala then sends this information to the hypothalamus, to the prefrontal cortex, and to a number of other structures on the hypothalamic region, and also sometimes to the brainstem at the hippocampus itself. And we're going to see pretty much all the outputs of the amygdala in a moment. Now, each one of these outputs essentially is responsible for a different function, and that's the reason why I was splitting them. And that's why it's also important to consider the limbic system as closely related to the basal ganglia system and also to the hypothalamic system. Now, one of the functions that the amygdala does is regulating your appetite, your feeding. So from those information that we've seen before, the amygdala sends information to the hypothalamus, specifically to the ventromedial nucleus of the hypothalamus, you can see here, and to the lateral hypothalamic nucleus, you can see here. There is a fine tuning of these nuclei regulating your essentially your need to be fed. So if the ventromedial nucleus is activated, you feel full. You don't actually want to eat anymore. There are experimental studies, so done only in animals, to actually target this nucleus for obese people to induce a sense of satiety to reduce their caloric input, essentially. If you hyperactivate the lateral hypothalamic nucleus, you actually want to eat more. The connection between the amygdala and these structures happens through the stria terminalis, which is a bundle of white matter with a similar course to the fornix, but it goes around the thalamus and is sitting pretty much here, and the so-called ventral amygdalofugal pathway. The ventral amygdalofugal pathway might struggle a bit to find it in books. Essentially runs from, this is the head of the hippocampus, so this section should be a bit more anterior, but let's say it's here. Runs essentially beneath the basal ganglia. There's going to be a picture showing this later on. This connection between the amygdala, which is a fine regulator of emotions, as we will see, and this area also explains why, when you stress, you might not want to eat. In some people, this is what happens, or like in my case, you want to eat a lot, and this is kind of explained by this neural connection that you see here. Another function that the amygdala does, it regulates your sexual arousal. So from all the information that we've seen before, so inputs, visual inputs, auditory inputs, and somatosensory inputs, you are in the presence of the love of your life. Your brain sends information to the amygdala through the pathways that we've seen before. The amygdala then sends information to the hypothalamus, again, through the stria terminalis and the fornix, probably also through the amygdalofugal pathway. The hypothalamus specifically activates two nuclei, the periventricular nucleus and the medial preoptic nucleus. What do? They increase the production of oxytocin, and they increase the production of testosterone. So your sex drive goes up. And this is essentially how the amygdala works in sex arousal. Okay. Another very important pathway is the one responsible for reward and behavior. Now, again, all the information that we've seen before, all the inputs, visual, somatosensory, visceral inputs as well, coming from the brainstem, activates the amygdala. The amygdala, in this case, have two outputs. One output goes to the septal nuclei through the same pathways that we've seen before, ventral amygdalofugal pathway, stria terminalis, fornix. Septal nuclei and lateral hypothalamus activate an area of the brainstem that is called the ventral tegmental area. This ventral tegmental area is the primary source of dopamine. I'm not sure if you've seen that meme online. There are two molecules that are responsible for pleasure in our brain. One is dopamine, the other one is serotonin. The serotonin pathway is a different one. This one is the main dopamine pathway. So the activation of the ventral segmental area causes two things. First of all, activates back in a feedback loop the amygdala, but also activates the nucleus accumbens. The nucleus accumbens is sitting, if you remember, close to the paraterminal parafactory gyrus in the frontal lobe, very basal, beneath the splenium of the corpus callosum. Sorry, beneath the head of the corpus callosum, very close to the rostrum, at the level of the a bit lower than the septum. The nucleus accumbens activates the prefrontal cortex. Whatever has caused you to get your amygdala very excited is now kicking. Your prefrontal cortex has this boost of dopamine and goes, oh wow, I really like that. And so it feeds back to the ventral tegmental area and asks for more dopamine, and so on. It feeds back to the ventral tegmental area through the mesolimbic pathway, the nucleus accumbens and the mesolimbic pathway. And so this reward pathway goes on and on and on and asks you for more. This happens when you are tasting something very good, or you are tasting something very good that is a bit illegal. So this is how you become addicted to drugs, for example, heroin and cocaine, pretty much all the addictive drugs that you know about, alcohol itself. What is important to remember about this pathway is that there are two very important white matter tracks, which are the mesial forebrain bundle. There's a lot of confusion on pretty much all the books and papers that that I've seen online. The mesial forebrain bundle essentially starts from the nucleus accumbens, receives a lot of information from the of other bundles from the lateral hypothalamus and from the amygdala, and ends up in the brainstem, crossing the hypothalamus essentially. This pathway of the mesial forebrain bundle is called the mesolimbic pathway. The connection, the direct connection between the brainstem and the prefrontal cortex, which releases dopamine to the prefrontal cortex, is called the mesocortical pathway. Okay, so this is the reward pathway. I couldn't find a better scheme than this. This is obviously a brain of a rat, but it's just to give you kind of an anatomical idea of how it works. So this is the olfactory bulb of a rat. This is his brainstem. You have the peduncle-pontine nucleus, which, let's put it aside for a second, but essentially is also involved in this pathway. But the main nucleus that I want you to remember is this, the ventral tegmental area, which is sitting in the brainstem, and this fascicle running here is the mesial forebrain bundle. This is the mesolimbic pathway. The direct connection of this nucleus with the medial prefrontal cortex is the mesocortical pathway. There is also, if you remember, I mentioned earlier on in the olfactory area, in fact, about the olfactory pathways, there is also a pathway involving the habenula. Now let's focus on this for a second. This is very controversial and it's very debated, actually. I found some sources discussing it, and they also say that there is a lot of studies going on about this. But essentially, this is how it works. This pathway is kind of the anti-reward pathway. So you remember I mentioned before the olfactory nucleus get in contact with the septal nuclei. The septal nuclei, through a very tiny bundle that we can actually see on the wall of the third ventricle, which is called the stria medullaris thalami, are connected with the habenula. The habenula is a region sitting on top of your pineal gland. It's a very small region, and there are two nuclei, a lateral habenular nuclei and a medial habenular nuclei. The habenula sends information through a small, very small white matter bundle, which is called the fasciculus retroflexus, send this information to the brainstem, specifically to the ventral tegmental area and to the rougher nuclei. Through the same pathway that we've seen before, the mesial forebrain bundle, this time around, the dopamine release is actually reduced. So through this pathway, there is, which is not the only one, by the way, so it's not the only one controlling dopamine, but it seems to be an important one. The dopamine release is opposite to the pathway that we're just seeing here, okay? The dopamine release here is reduced. What is also interesting to know is that the habenula has a potential connection with the rest of the brainstem, with an area of the brainstem called the area postrema, which is responsible for vomiting and nausea. This connection happens to a fasciculus called the dorsal longitudinal fasciculus. Again, this is debated, but I found some authors discussing this. The hypothesis between this pathway, regarding this pathway, is that you feel an unpleasant smell and you feel nauseated, and this is probably why. It's not the only one. The area postrema, the main input of the area postrema is from the solitary tract nucleus, which is coming from the vagal nerve. So that's probably the main input. But there is debate about the fact that there might be another input coming from the habenula. And these are the schemes essentially summarizing what I just told you. This is the stria medullari thalami. This is the pineal gland, corpus callosum, lateral ventricle, third ventricle. You got your medial labenula signaling down to the ventral tegmental area. This ventral tegmental area then reduces the dopamine release at the level of the nucleus accumbens and the prefrontal cortex. Now, almost done as well. Another function of the amygdala. So what we've seen so far, sex, feeding, and reward pathway. The flight or fight pathway. Now, this involves your autonomic system. So the amygdala, with all the connection that we've seen before, remember, so posterior association area, visual, somatosensory, auditory, and a number of other connections. Amygdala receives information about your external environment. I think it's easier to do this by doing an example. So let's say that you are walking down the street and an unleashed, giant, very aggressive dog comes in your way. What happens? Well, you get pretty scared, I suspect. I would. And so this pathway starts kicking in. So the amygdala, through the same connection that we've seen before, mainly the ventral amygdalofugal pathways and the stria terminalis, activates specifically this nucleus of the hypothalamus. Now, the nuclei of the hypothalamus of the the amygdala, activates the autonomic nervous system, specifically in the example that I've given you, the sympathetic nervous system. Now, the nuclei of the hypothalamus responsible for the autonomic nervous system are actually very easy to remember. The anterior medial nucleus of the hypothalamus is the parasympathetic one. The posterior and lateral nucleus of the hypothalamus is the sympathetic one. So now let's see that let's say that the sympathetic nucleus is activated. What happens? So you got an activation that descending activation on a number of systems. Specifically, what happened? You seen that dog. This example concerns rage, but it's pretty much the same thing. Fear can pretty much is actually very similar. So going down to your spinal cord and then to the sympathetic chain, what happens? Your amygdala through this pathway is asking what is asking you? Essentially to either fight or to flight, so to run away. So I will need a lot of oxygen everywhere. So ask your heart to start pumping blood everywhere. The blood pressure needs to be risen. So vasoconstriction peripherally, blood shifted to muscle, mostly heart itself and brain. So blood pressure goes up. Liver, we need glucose to do this. We need to activate our muscles. So release a lot of glucose to actually help us. Adrenal gland, release a lot of adrenaline to help the system working again. And it's not represented here, but muscles increase, lungs, increase your respiratory rates so that we can get more oxygen, we can breathe better, and we can actually either run away or fight a bit better. So this is the system that gets activated through the amygdala. Obviously, the opposite reaction to that, when you're very relaxed, you have activation of the parasympathetic nucleus, which essentially is gonna sort the absolutely antithetic reaction to that. So you have decreased heart rate, decrease of blood pressure, a generalized feeling of being relaxed. Now, another function of the amygdala, so decision making. This is actually very complicated, but I try to make it as easy as possible, as comprehensible as possible. So again, all the inputs that we've seen before, the amygdala through the same pathways that we just seen, this time around activates the thalamus, specifically the nucleus that I mentioned earlier on, so the mediodorsal group. The mediodorsal group, specifically the parvocellular area of the mediodorsal group, so the area of the mediodorsal group that is made of small neurons, activates the cingulate cortex. The cingulate cortex sends information to the prefrontal cortex and to the posterior association area. The posterior association area feed back to the amygdala in itself, and you have this loop ongoing that helps you essentially make decision. This is how it works. There are some, on this paper is very interesting because it highlights that this particular, the mediodorsal group also receives inputs from the basal ganglia and from the superior colliculus, which, the superior colliculus, which activates the frontal eye field. So when you have a decision to take, essentially what you see is the activation of this pathway, and also a big activation of the prefrontal cortex and the anterior portion of the cingulate gyrus. The cingulate gyrus is probably the most important structure here to help you making decision. What you have in people with dementia or with advanced hydrocephalus is that they're incapable of making decision, and they're actually incapable even of orientating themselves in their environment. And the reason for that is because this pathway is severely affected. Very related to this pathway is the pathway involved in attention. The amygdala, again, after receiving all the information from the cortex that we've seen before, through the ventral amygdalofocal pathway and the stria terminalis, activates this nucleus, which you can find named either as nucleus basalis or basal nucleus of Meynert. This is the main source of acetylcholine in the brain, and it sends informations everywhere in the cortex, mostly to the cingulate gyrus, but also to the prefrontal cortex and to the parietal lobes as well. And what it does, essentially, this pathway is the one responsible for your focus. So it activates when you actually need to be concentrate and pay attention to what you're seeing or listening to. So I really hope that this pathway is still active as I speak now, because I suspect it's a bit switched off after almost 50 minutes of lecture about the limbic system. What is important to remember is that some of these, a portion of this pathway is also activated by the brainstem through the same ventral tegmental area and to the same pathway that we've seen before. As you can imagine, all these pathways that I've shown you are actually kind of acting synergically depending on the external stimuli that you're having. And that's why the limbic system in itself is so complex even to describe, because they actually kind of work synergically together. And this is an anatomical summary of what I've just told you. In this particular example, the stimulus that is responsible for activation of this pathway is an auditory stimulus. So if you're listening to a podcast or to music or whatever you want to put your attention to it. What I found very interesting about this scheme that I found on this paper is that some of the connections are actually pointed out they're actually unknown, and there's a very big question mark here, which gives you an idea of how much is still to clarify about these topics. So with this pathway, we actually finish the overview regarding the amygdala and pretty much about the whole limbic system. What I wanted to show you here is kind of a summary of some of the bundles that we have seen and some of the nuclei that we have seen. So you can see here in this schematic representation, this is the amygdala. On the, unfortunately, sorry, this is the left side and this is the right-hand side, but you have an idea. The asterisk here is the amygdala. And what you can see here are, so this is the lateral olfactory stria, sorry, sitting here. This is the olfactory bulb. This is the medial olfactory stria rising up to the septal area. This is the nucleus accumbens. This is your amygdala. And what you see here is the ventral amygdalofugal pathway with the diagonal band of Broca that we mentioned earlier on. On top of which it sits the anterior commissure that we're seeing here. This is your corpus callosum, and this is essentially the septal area and your ventricle. This is a view from the base of the brain, okay? So this beautiful dissection is the only paper when I actually managed to see this, when you actually see the ventral amygdalofugal pathway and the diagonal band of Broca. As you can see, deep inside here, you have, this is your thalamus. Deep inside here, you have the third ventricle and the hypothalamus. So you can see how closely related these structures are. What is also interesting to see is that from the amygdala, you have this bundle running here, receiving connection from the nucleus accumbens. This is the mesial forebrain bundle, and it gets into close contact with this area here, which is the brainstem. Has been removed here, but the midbrain and the ventral tegmental here are actually sitting here, just below the thalamus, and this is your thalamus. On top of the thalamus, you have the stria medullaris thalami, a bit on top of which you could see theoretically, you cannot see it here, but you got the stria terminalis thalami, which is between the thalamus and the head of the coded nucleus. A few references for you to read if you want to learn more about this, and some websites that I've actually used to help me to refresh a bit more the limbic system, because trust me, guys, it's really complicated. It's something very, very difficult to actually refresh even for me, to be honest. So this website describes very well the limbic system is divided in several sections. This is a beautiful, I would say, introductory video from Penn State University. And this is another presentation from an Indian physician that you can find online. And here are some of the papers that I mentioned for the purpose of this lecture here. This is it from my side. Happy to take any question.
Thank you very much, Giulio. That was excellent, especially and I'm sure everyone in this group learned. You can happily unmute yourself.
Sorry, Nicola, I think I've lost some of the bits.
Or just stick your hand up.
Nicholas, it's very hard to hear you.
You got the QR code.
Oh, yes, absolutely. If there are no questions, I'll show you the QR code. That's the one.
Yeah, and if everyone in the group could please fill in the feedback for Giulio. It's clear he went into a lot of effort for the presentation, and it's used towards your certificates as well.
People are sending you claps, Giulio.
Oh, thank you very much, guys. Thanks a lot. Thanks a lot. I actually enjoy preparing this. I mean, it was difficult to concentrate everything in 1 hour because it's such a complicated topic, I'm telling you. But yeah, I'm glad that it was appreciated, and I hope it was clear. Sorry if it felt messed up, but I tried to be as orderly as I could.
Sorry, I don't have a question, but I can't see the QR code on my screen. Is that just me?
No, sorry, I'll put it back.
No, that's...
Here we go. Can you guys see it? No, sorry.
No, it's not come up yet.
How about now?
No, I think the screen perhaps is frozen.
Okay, let me refresh it. I'll stop sharing and I will start again. Just give me a second. Sorry, guys, for this. How about now?
Yeah.
Fantastic.
Yep, Ginevra, would you like to ask your question?
Yes, thank you. This presentation was really interesting. I was wondering if there are any evidence, it's my curiosity, if there are any evidence of a connection of the limbic system with dreams, because I know that dreams are connected with hypothalamus and amygdala too, but and also prefrontal cortex. So I was wondering if there is a connection actually or not.
Excellent question. So the answer is yes. I would struggle now to be absolutely precise about that. But what I can tell you is that there is a very spread out network in the brain that is called the default network. You can look that up. That is very heavily activated when you are not focusing, and also when you're sleeping. Is essentially a network involving the parietal lobe, specifically the posterior association area, the prefrontal cortex, although in a different way than we're seeing now, and portion of the thalamus, especially the pulvinar and the posterior portion of the thalamus. And there is definitely a connection with the amygdala through the stria terminalis and stria medullaris. I cannot be more precise than that, but I can definitely look into that. So what does it do? Essentially, this particular default network is the one activated where you're during your REM cycles, for example, and when you're not focusing. So let's say you're taking a walk and you're thinking nothing, literally nothing. You know, where you let your train of thought go. What it does is that essentially these networks mix together information from different memories, and that's why it receives information from the hippocampus as well. Actually, I forgot to mention that. Different memories, different sensory stimuli, and different information stored in your cortex, and you literally mix them together. And that's why during your dreams you have this very weird association of thoughts, like you have flying cows, or well, at least I do. you see flying cows, or I don't know. You imagine being in situations that are clearly not real, like flying, for example, or whatever. So, and it's also a network that is activated when you are actually particularly creative. So, it might happen to you. I mean, it happens to a number of people a lot of times that you're not really focusing on anything, and all of a sudden your brain associates to idea, and you have actually a great idea that you can apply to your daily life or to your job or to your studies as well. The reason for that is because this is also called the creativity network, and again, it's activated also in the REM cycle. The reason for that is because all of a sudden your brain is actually associating things that normally you wouldn't associate if you were focusing, and so. And that's why sometimes some people are more creative when they're distracted, because this is exactly what happened. There's a professor of neurologist at Imperial called Professor Payam, I think. I'm not sure, but anyway, we were having this conversation during functional operation, where he was saying, using this example in a soccer player. And he was putting the example of Cristiano Ronaldo and Messi, and he was telling us that Cristiano Ronaldo is a very frontal guy, and he uses his amygdala and his attention pathway a lot to be really focused. And so that's why he continuously looking for schemes and stuff to do when he's playing. While Messi is absolutely the other way around. 98% of the time he doesn't do anything when he's playing, but then all of a sudden he comes out with that particular action that actually twists the game completely in his team favor, because it's more a person activating the default network. So yes, the short answer is yes, the connections are there. I can't really remember the precise connection, but if you want to contact me via email, I can look into that and give you a more concise and a more precise answer.
Thank you for your answer. I will contact you by email, maybe, if I can have it, please.
Yeah, sure. You can mail the guys who organized this lecture. Absolutely. I would tell it to you, but my surname is impossible to spell. So if you ask Nicola or whoever, I'm very happy to reply.
Yeah,
Thank you very much.
Amira, do you have a question, or was your hand up from the previous QR code?
No, I have a question, I promise, but it's one and then I will shush, I promise. I just wanted to ask, when it comes, you mentioned in advanced hydrocephalus, decision-making is impaired. I just wanted to ask if that would obviously return to normal once the hydrocephalus has been resolved, or do you see that sometimes it's damaged beyond repair? Because I know you mentioned that certain parts of the limbic system are vulnerable to hypoxia.
Yes, absolutely. So the main portion that is very vulnerable to hypoxia is the hippocampus. So when you have a brain concussion, you have this retrograde or anterograde amnesia because of the hippocampus.